series of tubules. It's going all the way, or lots of them having bazillions, I don't know the exact number, bazillions of germ cells. And so imagine you take a cross section of one of those tubules, right? And then we're just showing one side of it. So imagine the whole tubule going around like this. The lumen, the empty part in the middle, that's where the spermatozoa are going to be released to become sperm. Okay, so we're just looking at the side, essentially like think of that one wall. That's the seminiferous epithelium. What's really kind of cool about this is the way these cells are arranged. So this is the exterior going towards the exterior of the tubule. These are arranged so that the mitotically active spermatogonia are on the outside, furthest from the lumen. Then as you go through meiosis 1 with the primary spermatocytes, meiosis 2 with the secondary spermatocytes, and then the round spermatid and spermatozoa, it's going from the outside towards the inside. So it's this nice progression of events that you can see. Now these are the things that I want you to learn. Some could be review, some might be not review. Primary spermatocyte, ogonium refers to mitotically active. We see spermatogonium here. Oogonium would be a mitotically active female germ cell in the embryo. Okay, this is all taking place postnatally. Primary spermatocyte, we'll see primary oocyte in the next slide. Secondary spermatocyte, secondary oocyte. Now there aren't any spermatids in the female. We have, at this point, we get a little bit of divergence between male and female. But take a look at, as a reminder, you'd be able to tell from the chromosome complement the number of chromosomes, amount of DNA, when meiosis 1 has been completed, when meiosis 2. Now this is the maturation and it's kind of specialization in spermiogenesis. Just know that that takes place. You don't have to know details, but it's really kind of a fascinating thing that occurs here. All of the histones in the chromatin, now I can't remember, I think it starts at the spermatid stage, but it's definitely completed by the stage of spermatozoa. All the histones are replaced by kind of an equivalent protein structure called protamines. But that allows that real tight compaction of the sperm nucleus, right? Because basically, sorry guys, but basically the sperm is just DNA packed into a tiny little compartment with a tail so it can swim. Okay? But that compaction has to take place at a much higher level than you would see in any other nucleus. Okay, so here's now, I'm going to break this down in the next couple of slides into the first part and the second part. But again, I do want you to know these terms. You don't have to know. So there's all kinds of amazing things taking place with the somatic cells, the follicle, so that surrounds each of the oocytes. You don't have to know that terminology. But just know that it is interesting. So the first, I just wanted to show you this because I love the drawing. It's from the 1940s. But it just gives you a perspective on the size difference between an oocyte. And this is fully grown. So these are the somatic cells that surround the oocyte. This is the oocyte with its massive cytoplasm. There's the nucleus. But drawing it to scale compared to the sperm. Little teeny tiny guy here, big massive cytoplasm and nucleus in the oocyte. Now this is that same figure, and this is taken it's from one of the textbooks. This is the top part of the term, oogonium, mitotic in the embryo, primary and secondary oocyte. So we're just looking up through the first meiotic division. There's the first polar body, secondary oocyte. So meiosis 1 has been completed. This is actually what gets ovulated. And that's that animation, or not an animation, video. What I wanted you to see in that, though, let me find it again, because it is kind of cool. There's some things that are happening that you might not. Anyway, what I wanted you to appreciate is that before meiosis 1 has been completed, the chromosomes are located here. What they have to do is move from the interior. They have to be positioned right next to the membrane. And that's controlled. It's not just random, it's controlled, and it's got to position itself so that the mitotic spindle is oriented, so that the chromosomes that are segregated into the polar body are going in the right direction. Again, that's controlled. So one example of more things going on at the level of the cells than what you might imagine. And there are scientists that, not so much, I don't think it's been done with humans, but with mouse. You can actually do RNAi, RNA interference, and inject into these oocytes RNAi against specific genes and disrupt this process. There's also some evidence, and this is from humans, that there is preferential segregation of the chromosomes into the polar body, maternal versus paternal, which is really kind of interesting. I'm not sure how common that is, but there's evidence that it does happen. Okay, then completion. This is ovulation, so this is where we were from the previous slide. Secondary oocyte, first polar body. This is what gets ovulated, and then it doesn't complete meiosis 2 unless fertilization is taking place. Okay. Now, there's a lot more things happening here. So we're going from fertilization. Here's the two pronuclei. This is setting up the first mitotic spindle for that first mitosis that takes place from the zygote. There's the two polar bodies. Okay. Now let's start over here. Here's the egg nucleus, polar body 1. The zona pellucida is a special structure that forms around each oocyte. The genes that encode this are oocyte specific. It's in part because of the zona that we have a species-specific fertilization. That there are proteins in the sperm that have to recognize proteins in the zona in order to be able to penetrate. The zona pellucida surrounds each oocyte. Now we'll see an animation of this in a few minutes, but once the sperm has entered into the oocyte, we get what's called a cortical reaction. There's a change that takes place on the exterior around the oocyte once it's been fertilized. This cortical reaction doesn't occur if there's no fertilization. What do you think that cortical reaction might be mediating? Remember, it's on the outside of the oocyte. It's right after one sperm is entered. Yeah, because we don't want polysperm if there's multiple sperm. Okay, now occasionally a second sperm will fertilize. That's very rare, and there have been some live births with that. Okay, we'll see what happens in a second. But then this also triggers the second mitotic division. So we'll see how fertilization triggers not only the cortical reaction, so there's not polyspermy, it actually is affecting the cell cycle progression of meiosis. Okay, so there's a male pronucleus, there's both the polar bodies, there's the female pronucleus, and then now we've got, you don't see it here, but the two pronuclei will fuse, and you get that first metaphase, and then mitosis occurs. Let's see some of the things that are taking place right after fertilization. Oh, okay. So I just want to warn you of this video. I actually took it from one of those textbooks, and I just copied it over here. We'll see a couple more on Thursday. I posted these. You should be able to start the video. There's no audio. Now, this starts out with an anatomically correct naked female. So just to warn you. Cover your eyes. You should know what's happening with your own body as well as for this class. So now we're gonna see, so there's the uterus, vagina, uterus, ovaries, fallopian tube. I'm gonna zoom in and look at some of the oocytes. These are the actual follicles. They're in different stages of growth. This is one that's about to ovulate. That's a secondary oocyte. Rupture of the ovarian, ouch, it literally does break through. Now some cells do come attached along with the oocyte, and it just kind of travels down into the oviduct, fallopian tubes. And it reaches the top of the uterus. Now we're going to zoom in. There's that nice metaphase spindle with the first polar body, and then the purple cells are somatic cells that surround that oocyte, and they just kind of travel along. Now we're going to have the guys coming along. There's the bazillions of sperm swimming along, moving from the vagina up through to the uterus, and they're going to get, most of them will get all the way to the top, and really it is a race, so that one lucky guy, one lucky sperm, there's 3 of them here, 4 of them, only one will make it. And it literally does release enzymes. It literally does penetrate into the zona and other structures there. Once it reaches this point, it's about to enter into the oocyte, and then the tail is going to break off, because we don't want to have the tail inside the oocyte. That, then, we won't go, there's no description of the details of this, but just at that point, the protamines that I told you replace the histones, the protamines would be replaced by the normal histones, and then you start getting decondensation of the male. Oops. Sorry. Okay, now once the sperm head enters, that's what will trigger the cortical reaction taking place among them. And we now want to have every other cell not to have. There's the cortical reaction. The block is the collagen. Now, what that triggers, and so we do nothing, that triggers the onset of anaphase, which I believe is closest to collagen. And so it triggers the exclusion of the second polar. Then we get that spindle dissolve, then they fuse and form the first mitotic spindle. So we've gone from a meiotic spindle to a mitotic spindle, and you might be thinking they're the same. There's a real important difference. Go along with some of the other things. Okay, just in case you wonder, just so you're trying to get an idea of what happens with baby fertilization. A woman is treated with a whole bunch of hormones that can raise multiple oocytes at the same time. The first culture, the sperm from the man is collected, they mix together, they look, the technicians in the IVF clinic, this is reproductive clinic, with like the cell therapies, cells in culture, and we watch the ones that get fertilized, and then if they watch for the first time like those, you can say, please, if they go according to the correct time, and the cells look normal, they'll pick up the nurses looking normal, and then keep going. I think this is a two-day procedure. Something like that. And also, of course, any healthy looking embryos could be frozen. So you probably all heard the chorus where it's at the freezers, and some, this is a big difference in things malfunctioning. It's a big deal. Okay, just to give you an idea, you can look at the level of statistics if you're interested. This is a 2017 report, but it's from data collected up through 2014. This is the incidence, the increase in incidence of assisted reproduction cycles, live births, and infants born. And sometimes there's multiple infants born, because they sometimes will either implant just one embryo or multiple embryos. But it's increasing. Now remember this, the technology has been around only for 40 years. Louise Brown, the first guy in the baby, rescue baby, she turned 40 years old last summer. Okay. And this, I don't even know, I mean, the incidence now, it's just going up and up and up and up all around the world. So, this is a great quote to take from this review article. Egg to the embryo, this is really important. And there's a lot of things that have to take place, both at the level of the cells and at the level of the genome and what's going on inside these cells. So this is the two pictures I showed you previously. At these cleavage stages where the zygote goes through the first mitosis, then there's the second mitosis, then the third mitosis, you can call each of the cells is genetically and at levels of all function and gene expression, everything, they're identical. There's no differentiation taking place. They're literally daughter cells. You can refer to these as being blastomers, that's the correct blastomeres. And that name comes from what will happen further down the line is a formation of a structure of an embryo called the blastocyst. So blastomere is an individual cell. You have 2 blastomeres, then 4, then 8, and 16, and 32. What we'll see is once you get, I think you can take 4 blastomere, one of these at the 4 cell stage, definitely at the 8 and 16 and 32 cell stage, you can actually remove one of the blastomeres and do DNA analysis on it for pre-implantation genetic diagnosis. It's done all the time in these assisted reproduction clinics. Okay, this is a figure from that review article. Don't worry, you don't have to know all this. Look at the slides because I've got, now I'm going to step through what's happening here. These are what I want you to learn. So we've got the sperm entering. Before the sperm has entered, though, remember, we've got the oocyte is arrested in metaphase. Okay. This structure, APC, that stands for anaphase promoted complex. What that does is control an enzyme. It's actually a complex of proteins. It indirectly controls the enzyme that will cleave the cohesin that holds those cystine molecules together. So if APCC is being inhibited, you're going to stay in metaphase. Once you have active APCC, then you get separation of the sister chromatids, and that's the onset of anaphase. So that's what we have here. Here's the endoplasmic reticulum. There's calcium inside the cells. You don't have to know what PLC is, but it's a phospholipase C. That's actually carried by the sperm that enters into the oocyte. What that does, and I'm not showing you all the details. If you want to know, look in the original figure in the review article. But what that does, along with activation of a protein here at the membrane, triggers the release of calcium that has been stored in the ER. Okay. Now that release of calcium does a number of things. Indirectly, it inhibits the inhibitor of APCC. Once you inhibit the inhibitor of APCC, then you get active APCC. You get the completion of meiosis. Okay. Calcium does a couple other things. That triggers cortical granule exocytosis, the cortical that block is polyspermy. Translation of maternal mRNAs. That cytoplasm of the oocyte has had a lot of mRNAs that have been transcribed, but there's an active process for stopping the translation of the vast majority of them. So part of the importance of the cytoplasm of the oocyte for the early embryo is that shortly after fertilization, those maternal mRNAs will get translated. And this is before any of the genes in the zygote or embryo have been activated. So at the very early stages of embryonic development, the only gene products that are there are from the mom. That's the basis of many maternal effect mutations. In addition to initiating translation of these stored maternal mRNAs, you have that transition from minuses to micrones. Okay, I'll just show this, but this is where we'll get started. This is triggering APCC, just to remind you of what's happening. APCC activates, separates the enzyme that will cleave the cohesins that hold all this okay. If you came in late, just so you know, and we'll do this regularly, there's a attendance sheet. Just check your name. If you were here today, don't check your name.